Hi students, we'll continue with the study of biomolecules. We'll just have a quick recap of our previous class. In the previous class, we have discussed about Dharwad structure of glucose, later structure of fructose. Then we studied three disaccharides. First one is sucrose, its Dharwad structure, which is composed of alpha D plus glucose and beta D minus fructose with a C1C2 glycoside linkage. Second disaccharide we studied in previous class is maltose, which is a polymer of two alpha D plus glucose units in which C1C4 glycoside linkage is seen. The third disaccharide is lactose, which is composed of beta D galactose and beta D plus glucose, wherein the C1 of galactose is linked to C4 of glucose. Again, we come across C1C4 glycosidic linkage. Okay, so this much we have discussed in our previous class. Today we'll discuss polysaccharides. So already you know what are polysaccharides. Polysaccharides are the carbohydrate which undergo hydrolysis to produce more than 10 monosaccharide units, isn't it? So these contain a large number of monosaccharide units. I said no. So these undergo hydrolysis to produce more than 10 monosaccharide units, and these monosaccharide units are joined together by glycosidic linkage. And these polysaccharide, they act as a food storage as well as structural material. So we'll discuss three polysaccharides today. First one is starch, second one cellulose polysaccharide, and the third one glycogen. Okay, let me begin with the first polysaccharide, that is starch. This starch polysaccharide, it is a main storage polysaccharide in plants. Okay, the starch is stored in the plant, since it's called a storage polysaccharide. It is most important, prime important food source, dietary source for human beings. Our food mainly composed of what? Starch. Rice contains starch, our staple food is composed of what? Starch. Therefore, it's an important dietary source for human beings. The high content of this polysaccharide, starch polysaccharide, is found in cereals, roots, tubers, and in some vegetables. So chemically, starch composed of what? It is a polysaccharide composed of many monosaccharide units. Which monosaccharide units? Alpha D plus glucose monosaccharide units. Okay, it's a polymer of what? Alpha D plus glucose. Here I have mentioned only alpha glucose. It's a alpha D plus glucose because glucose is a dextrorotatory. Always positive configuration will be there. And it consists of two components. So starch polysaccharide is a polymer of alpha D plus glucose units and composed of two components. Those two components are number one amylose component and number two amylopectin component. Starch polysaccharide has these two components. Okay, we'll discuss these two components now. So this may be asked for 1 mark. Okay. Well, we'll see the differences between or study of both components, amylose and amylopectin. These two have some differences. First of all, the amylose component is water soluble, whereas amylopectin is water insoluble. And amylose is composed of what? 15 to 20% of a total starch. Okay, if you consider starch as 100%, out of 100%, 15 to 20% will be, that means a lesser percentage of amylose component will be there, whereas amylopectin component will be more. How much? Around remaining 80 to 85% of starch is composed of what? Amylopectin component. Starch is a very long unbranched chain. It's a straight unbranched chain of what? Alpha D plus glucose unit. Okay, polysaccharide contain many alpha D plus glucose unit and they form a very long chain. The chain is not branched, unbranched chain. Whereas amylopectin is a branched chain polymer. Okay. This amylose composed of around 200 to 1000 alpha D plus glucose unit. Okay, upon hydrolysis, amylose gives how many monosaccharide alpha D plus glucose units? 200 to 1000. Okay, and these 200 to 1000 alpha D plus glucose unit, they are joined together by, held together by a bond, a link. That link is called glycoside linkage. This we have discussed in previous class. No, the two monosaccharide are held together by a glycoside linkage. In amylose, the glycoside linkage is present between C1 C4. That means C1 of one glucose links with C4 of another glucose unit. Therefore, C1 C4 glycoside linkage we see in amylose component. Whereas amylopectin component also composed of 200 to 1000 alpha D plus glucose units only, and they too are held together by C1 C4 glycoside linkage. So apart from this, they have a branching. And that branching occurs at a C1 C6 linkage. So C1 C6 glycosidic linkage is a branching linkage, whereas C1 C4 is a straight chain linkage. Okay, we'll see structure so that it will be clear to you. Okay, got the differences between the two components. Amylose is water soluble, amylopectin water insoluble. Amylose composed of 15 to 20% of starch, whereas amylopectin composed of 80 to 85%. Amylose is an unbranched chain. Amylopectin is a branched chain. Amylose composed of C1 C4 glycosidic linkage. Amylopectin also composed of C1 C4 glycosidic linkage. Along with that, C1 C6 branching is there. Okay, this concept is important for 1 or 2 mark. We'll see the structure of amylose first. So this is amylose structure. Okay, in this structure you can see 3 alpha D plus glucose units which are held together by a glycosidic linkage. I said starch or amylose as well as amylopectin, they are composed of how many? 200 to 1000 alpha D plus glucose units? No, but here it's not possible to draw 200 300 structures. So therefore, in order to show the structure of a polymer, we'll write a partial structure. Okay, so how to write this? So we have this alpha D plus glucose unit, so wherein HOH was there, but it's involved in a linkage. Here HOH was there, ado link agide already. Then second carbon HOH, OHH, HOS, CS2OHH. This we discussed in previous class. No, okay. This one alpha D plus glucose links with another alpha D plus glucose where carbon number 1 and carbon number 4. You can see the pointer there. So carbon number 1 and carbon number 4 are involved in a link, and this linkage is what? This arrow mark linkage is alpha D plus glucose, sorry, C1 C4 glycosidic linkage. Again, this glucose of a chain links with another neighboring glucose again through the C1 C4 glycosidic linkage. This links with neighbor alpha D plus glucose through C1 C4. That links with one more through C1 C4. Here this molecule links with the previous one. So like this, the chain continues how many times? Many times, 200 to 1000 such units will be there. So in order to show the polymer, so you can see pointer ಕಾಣಿಸ್ತಾ ಇದೆ. So this zigzag mark or a small bracket subscript n we will also write. That indicates that this type of a structure repeats n times, many times. Okay, so at last you need to show this carbon oxygen dash zigzag mark, here carbon oxygen zigzag mark. So this indicates that the chain continues many times, n times. So now this is amylose structure. Very simple. You need to draw 2 to 3, or even we can draw 4. Here I have drawn 3, no, 4 also you can write. Then show C1C4 glycosidic linkage. So it will be structure of amylose component. We'll see amylopectin component what structure. In the second structure, you can observe there are two chains. This is one chain and this is another chain. Okay. These two chains are held together by again glycosidic linkage. First of all. So this alpha D plus glucose links with another alpha D plus glucose through this C1C4 linkage. So this links with previous one. That links with previous one. That means the chain gets continued. That means it is one alpha D plus glucose chain. So here below we have another chain. So this alpha D plus links with this links with this, and the chain continues. These two chains are held together by C1C6 what glycosidic linkage. So there is a branching. The terminal glucose molecule, the end glucose molecule of a one chain links with the another chain. Where exactly the linkage is there? So can you see this is carbon number 1 links with carbon number 6 of this glucose of another chain. So there is a branching present at amylopectin structure in a C1C6 what you call glycosidic linkage. Okay, so in previous examination the structures of amylose or amylopectin have not been asked, but the concept is important as far as competitive examination point is concerned. But if they ask in the examination, they are very simple. Where the job is write in case of amylose, write alpha D plus glucose, show C1C4 linkage and show continuation of a chain. In case of amylopectin, show two chains of alpha D plus glucose and show C1C6 glycosidic linkage. That's a branching between the two chains. Okay, so this is about the first polysaccharide starch, composed of how many components? Two. They are amylose and amylopectin component. We'll move to the next concept. Okay, before the moving to the next concept, let me ask you one question. And this question, look here, it's a question of CET July 2020. Now we are in August 2020 now. So we had CET examination for our seniors, for second PUC passed out students day before, that means last week. Okay, Friday we had a examination now. So in that Friday examination, CET examination, there was one question. The question is like this: C1C4 glycosidic bond or linkage is not found in, options are maltose, second option sucrose, third one lactose, and fourth one starch. Okay, I'll repeat the question once again. It's a CET examination, this year CET examination question. C1C4 glycosidic bond is not found in maltose or sucrose or lactose or starch. Okay, I will not tell the answer now. It's an assignment to you, a very simple one. You just pick one right option and mention in a comment box. Okay, it's a work for you. I'll see who will post very quickly. Okay, one more question. And this question is from annual examination question paper, the 36th C of March 2014 examination question paper contains the question: name the water insoluble component of starch, for 1 mark. Very simple. We have two components, amylose and amylopectin. Okay, again the comment the answer, right answer in the comment box of your Google Classroom app. One more question. This question was asked in a 2018 CET examination. The question is like this: the glycosidic linkage involved in linking the glucose units in amylose part of starch is? Okay, read the question carefully. The glycoside linkage involved in the linking the glucose units in amylose part of starch is option C1C4 beta linkage, C1C6 alpha linkage, C1C6 beta linkage, and C1C4 alpha linkage. Okay, here alpha linkage, beta linkage, that means if the two alpha D plus glucose are linked together, they are called alpha linkages. If two beta D plus glucoses are linked together, they are called as beta linkages. Okay, pick the right answer and try give the correct answer in a comment box. Okay, one more question. It is a NEET 2018 question paper question. Okay, you can see many questions have been asked in a previous year competitive examination. I have chosen very few. Okay, still many question papers contain questions on this carbohydrate concept. So very few I have chosen for you here. The difference between amylose and amylopectin is option number 1. Amylopectin have 1,4, that means 1,4 here means C1C4, 1,4 alpha linkage and 1,6 alpha linkage. Amylose have 1,4 alpha linkage and 1,6 beta linkage. Amylopectin have 1,4 alpha linkage and 1,6 beta linkage. Again 1,6 meaning is what? C1C6. And fourth option is amylose is made up of glucose and galactose. Okay, choose the right answer. Again, it's a very simple question. I am not explaining I am not telling the answer for these questions since the questions are very simple. Okay, if you find any difficulty in these questions, I'll definitely explain in my next class. Okay, since they are very simple, I am not going through in detail about these questions. So if you found difficulty, so you can comment in the comment box, raise your doubts in the comment box. Definitely I'll explain in next class, but these questions are assignment for you. Okay, we'll see the second polysaccharide. So first polysaccharide we have completed is starch. Second one, cellulose. Cellulose occurs exclusively in plants. Okay, that means animals do not contain cellulose. These are found in plants and it is the most abundant meaning here large amount. Okay, most abundant organic substance in a plant kingdom. Plants contain many organic compounds. Out of those many organic compounds, the more quantity of cellulose is present in a plant kingdom. It's the most abundant element and it's a prime constituent, predominant constituent of a cell wall of a plant cell. Where is the cellulose presence in the plant? Cell wall of plant cell composed of cellulose. Chemically, cellulose is a straight chain polysaccharide. Okay, composed of only beta D plus glucose unit. Here I have not mentioned D plus. Plus is not mentioned here. So this minus is not a sign. Okay, it's not a levorotatory sign. So into bracket if you write minus, it's a levorotatory. So this minus sign is not levorotatory. Okay, so here I have not mentioned anything means glucose is a dextrorotatory only. Beta D glucose meaning is also what? Beta D plus glucose. It is composed of only beta D plus glucose units which are joined together by glycoside linkage, where again C1C4. C1 of one beta glucose unit links with C4 of the next glucose unit. The Dharwad structure will be like this. Again, it's a polysaccharide now composed of many beta D plus glucose unit. Okay, how to write this? So write two beta D plus glucose units. Okay, show the continuation of the chain and link those two by what? C1, C4 glycosidic linkage. Okay, look at the structure. This is beta D plus glucose. So beta means so here OH, H will come, no? Okay, so it's actually structure of cellulose. So if you write the structure of beta D plus glucose, here whatever the round marking I have done, no? So there OH, here H, H, OH, OH, H, H, OH, then CS2OH, H will be there. It's a beta D plus glucose structure. Now, what happens here? So this beta D plus glucose links with another beta D plus glucose. So where exactly? Here OH was there in a glucose unit, and here OH group was there, and these OH groups, they undergo dehydration, loss of water molecule, and they link a glycosidic linkage here, and this glycosidic linkage is present between C1. So here C1 carbon will be there, C1, and here C4 will be there. C1 carbon links with a C4 carbon, and therefore this linkage is C1 C6 glycosidic linkage, and this chain continues how many times? N times. That means this beta D plus links with neighboring another, this links with neighboring another, like this. The chain continues n times, and this is the structure of cellulose, composed of beta D plus glucose unit with a C1 C4 glycosidic linkage. Very important for 2 mark. Okay. We'll see one question. This is Manipal 2013. It's a competitive examination conducted by the Manipal University for the admissions to their courses. The incorrect statement regarding cellulose is, so among these, which is an incorrect statement? Okay, so I'll skip this question. The question is wrong, so it is wrongly typed here, therefore I will not go through this question. We'll see that question later in the next class. The third one is glycogen. It's a polysaccharide, carbohydrate stored in animal body. Just recall your memory. Cellulose is stored in a plant kingdom, whereas glycogen is stored in an animal body. It's a glycogen. The carbohydrate stored in the animal body is glycogen. It is also called as animal starch because of 2 reasons. First one, the structure of glycogen is similar to amylopectin of a starch. Okay, amylopectin is a component of a starch, no, which is a branched structure. The structure of glycogen is similar to that branched structure of a amylopectin. Therefore, it's called as what? Amylopectin. No, sorry, animal starch. And amylopectin is a branched that you have discussed, whereas glycogen is highly branched. Okay, so first reason, since the carbohydrate is stored in animal body, called as animal starch. Second thing, the structure looks like a starch, hence the name animal starch. It is present in liver, muscles and brain of an animal body. Whenever our body, human body or animal body, needs glucose, the enzyme breaks the glycogen down to glucose. That means in our body, the glucose converted into glycogen, and this glycogen is stored in liver, muscles, brain. Again, whenever body needs glycogen, that glycogen converted into glucose. Structurally, the structure of glycogen is not there for your study. Okay, because it's a highly branched structure. We have the study of starch structure as well as cellulose structure, but not glycogen structure. Remember this thing about glycogen. the last concept of this carbohydrate biomolecule, the biological importance of carbohydrate. First of all, carbohydrates are a source of energy. These carbohydrates are important, essential for life in both plants as well as animals, because they provide energy. They form a major portion of our food. Okay, our staple food is composed of carbohydrate. This polysaccharide, the starch, whatever we consume as a food, the food undergo digestion. No, the digestion is what? The polysaccharide broken down into smaller component. This is a digestion. The enzymes hydrolyze the polysaccharide into maltose first. That means polysaccharide converted into disaccharide. That disaccharide converted into monosaccharide. That means complex food materials broken into what? Simpler food materials. This is a digestion. Finally, the glucose after digestion gets absorbed into the blood, and that glucose through the blood is taken to cells of all organs, where they get oxidized to carbon dioxide and water along with the release of heat. Okay, this paragraph explains how carbohydrate act as a source of energy. Okay, ಅದ್ರ ಬಗ್ಗೆ ಹೇಳ್ತದೆ. We take the food undergo digestion, produces a glucose moves to every cells through the blood carries the glucose, where the glucose undergo oxidation to produce carbon dioxide and water. During this process, oxidation of a glucose process, energy is released. Okay, look at this equation. It's the oxidation of glucose equation. C6H12O6, glucose in the presence of oxygen undergo oxidation to produce 6CO2 and 6H2O, carbon dioxide and water, along with the release of this much of energy. Delta H, change in enthalpy, is equals to minus 2850 kilojoules per mole of energy. That means oxidation of 1 mole of glucose produces 2850 kilojoules per mole of energy. It's an exothermic process, and this energy released during oxidation of glucose is used for doing work and maintaining body temperature. And excess glucose, okay, so whatever the amount of glucose energy needed by the body, that much of amount is released by the oxidation of a glucose. Excess glucose in our body is stored in the form of glycogen, another polysaccharide. So by this note, we can conclude that carbohydrate is a good source of energy. Second biological importance. Carbohydrate, the two insoluble carbohydrate polymer, for example cellulose, act as a structural material. So for example, cellulose in plants, no, it provides a raw material for many industries like textile, paper industry, etc. Okay, so this textile raw material comes from a cellulose. That means it acts as a structural material. So cellulose acts as a structural material. The third biological importance, simple sugars like ribose, beta D2 ribose and deoxyribose, and deoxyribose are the components of RNA and DNA respectively. Okay, these are nucleic acids. About this RNA and DNA we'll study at the end of this chapter. Okay, so detailed study is there. This is about what biological importance of carbohydrate. Okay, so with this we have concluded the study of first biomolecule, that is carbohydrate. So we'll have a quick recap. So in the first class we have started with a study of what are biomolecules, what are carbohydrates, definition, classification into 3 classes. Then we discussed monosaccharides, aldoses, ketoses, again trioses, tetroses, etc. Glucose dali, structure elucidation of glucose, open chain structure, Haworth structure, isn't it? Then anomers, alpha form, beta form. Then we had a study of fructose, its preparation, not preparation, open chain structure as well as Haworth structure. Sucrose, its Haworth structure, prepare what you call hydrolysis reaction, then inversion of sugar, maltose structure, lactose structure, polysaccharides only today's concept, starch, two components, amylose and amylopectin, cellulose, and last one, glycogen, along with a few biological importance of carbohydrate. Okay, so we have completed carbohydrate study now. In the next class, let me begin with the second biomolecule, that is proteins. Thank you very much.